Hello, good day. In this video, we're gonna talk about Fasciola hepatica. What is Fasciola hepatica? Fasciola hepatica, also known as the common liver fluke or sheep liver fluke, is a parasitic flatworm of the class Trematoda, phylum Platyhelminthes, that infect the livers of various mammals including humans. The disease caused by the fluke is called fascioliasis, also known as fascioliosis. Fasciola hepatica is distributed worldwide and caused great economic losses in sheep and cattle. It has been known as an important parasitic of the sheep and cattle for hundreds of years. From the definition of Fasciola hepatica, what happened is, once this parasite enters the body, this fluke actually infiltrates various parts of the body, including the blood vessels, gastrointestinal tract, lungs, and liver, depending on the species, and in human, another very common spot is the gallbladder and bile duct system. So as this definition suggests, this disease does affect human, but it most commonly found in cattle and sheep. Humans may be contract with disease, so now that we know the basic of Fasciola hepatica, let's take a closer look on how one can contract the disease. The life cycle of Fasciola hepatica starts when a female lays egg in the liver of an infected human. Immature eggs are discharged in the biliary duct and taken out in the feces. If landed in water, the eggs become embryonated and develop larvae called myracidia. A myracidium invades an aquatic snail and develops into cercaria, a larva that is capable of swimming with its large tail. So this is the life cycle of Fasciola hepatica. It involves a final host where the adult worms live, an intermediate host where the larva stages of worm develop, and a carrier entailing suitable aquatic plants. The process starts when the infected animals, cattle, sheep, or other animals defecate in freshwater source. So if we take a look on the image, we see what the basic life cycle of the disease looks like. So we're gonna begin here with the sheep and cattle. The ruminants of the typical definitive host. So from here, we have unembryonated eggs which are passed in feces. So this is actually a freshwater source and the eggs become embryonated in the water. Since the worm lives in the bile ducts of such animals, its eggs are evacuated in feces and hatch it into larvae that lodge in a particular type of water snail, which is the intermediate host. Once in the snail, the larvae reproduce and eventually release more larvae into the water. These larvae swim nearby aquatic or semi-aquatic plants where they attach to the leaves and stems and form small cysts or metacercariae. When the plants with the small cyst attached are ingested, they act as carrier of the infection. Watercress and watermint are good plants for transmitting fascioliosis, but encysted larvae may also be found on many other salad vegetables that we are eating. Ingestions of free metacercariae floating on the water, possibly detached from carrier plants, may also be a possible mode of transmission. So another mode of transmission is if humans go and drink water from the source, they may actually drink this little miceriae, and in this way they can also develop the infection. The cycle of events in infection. It finds the liver and starts eating liver cell. This happens only a few days after the initial contact with the parasite. Usually the larva spends a few weeks just browsing and eating the liver. Then it relocates to the bile duct where it begins its final stage and become an adult. It takes about 3 months for the metacercariae to develop into an adult. Adult are about 3 cm long and 1 cm wide. Adult females can produce up to 25,000 eggs per day. The pathology and liver damage. Little damage is done by juveniles penetrating the intestinal wall and the capsule surrounding the liver, but much more necrosis result from migration of flukes through the liver parenchyma. Worms in bile ducts cause inflammation and edema, which turns stimulate production of fibrous tissues in the walls of these ducts. Thus thickened, the ducts can handle less bile and are less responsive to needs of the liver. Back pressure causes atrophy of the liver parenchyma with concomitant cirrhosis and possible jaundice. In heavy infection, the gallbladder is damaged and cell walls of the bile ducts are eroded completely. Next, in acute infection, the immature flukes migrate through the intestinal wall, the peritoneal cavity, the liver capsule, and the parenchyma of the liver before entering the biliary ducts, where they mature to adulthood in about 3 to 4 months. During this time, the patient suffers from abdominal pain, anemia, hepatic tenderness, hepatomegaly resulting from edema, intermittent fever, jaundice, lethargy or lack of energy, nausea, prolonged high fever, secondary infection, and vomiting. Chronic infection may be asymptomatic or lead to intermittent abdominal pain. So if we take a look on this picture, we can see the leafy-like structure found in the various spots on the liver tissue. And this is actually the infiltration of the various fluke within the parenchyma of the liver. And it usually takes 3 to 4 months for them to develop into adult stage. The infection typically is diagnosed by stool samples, duodenal or biliary aspirate, antibody test, can detect 2 weeks after infection, ultrasound, visualize adult in bile duct, CT scan, and reveals barrow in liver. All patients with hepatic phase were treated using Bitoneal because it is highly effective, large dose, high cost, and long-term treatment period. Another one is triclavendazole. It is easier to use, 1 to 2 oral doses in 24 hours, and virtually 100% effective, or worse, it may lead to surgery. Control methods. Education. It is the cheapest and most effective way. Wash aquatic vegetable in 6% vinegar for 5 to 10 minutes. Better herding practices. Keep herds away from aquatic areas. Individual people can protect themselves by not eating raw watercress and other water plants, especially from fasciola. Another control method is using molluscicide to get rid of snail and other pests. And that's all. Thank you.